Hello, good evening everyone. Welcome to you the new End Biology webinar series presented to you by the Translational Outcome Research Group of the Department of Zoology, University of Calcutta. This new series aims to celebrate the latest advances in the field of life science along with its relatedness to other disciplines. I am Ronita and I will be your moderator for today's session. Before we begin, I would like to inform you that you can post your questions in the YouTube chat box and we shall moderate them at the end of the lecture. You are free to interact with each other and with the speaker. If you have any question or comments meant for the speaker or for us, feel free to email us and we shall address them to the best of our abilities. And you will also send us your feedbacks through the feedback link, which will be provided in the YouTube chat box near the end of the session. Our convener, Professor Dr. Ina Banerjee, has sent welcome greetings for our speaker and to all of us. So, we, on behalf of our madam, welcome Dr. Bidisha Sinha in this Ant Biology platform and are requesting you, madam, to please deliver your valuable speech to enrich all of us here. Now, I will give you a brief introduction of today's speaker. Dr. Bidisha Sinha is an associate professor, Indian Institute of Science, Education and Research, Kolkata. She did her postdoctoral work in the Institut Curie in Paris in the year of 2007 to 2011. Did her PhD in Life Sciences at National Center of Biological Sciences in the year 2007 and took her bachelor degree in physics from Benares Hindu University in Varanasi, India. Dr. Sinha has research interest in the theme of the lab is to study how membrane homeostasis is established in cells. Membrane fluctuations are measured using interference reflection microscopy to decode the map of membrane tension in cells and understand regulatory mechanisms. Dr. Sinha took some very prestigious awards and honors from Wellcome Trust, DBT India, Alliance Intermediate Fellowship, and Ramanujan Fellowship in the year 2013. We are glad to host her today and excited to hear from her. So, madam, I welcome you once again, and the platform is all yours.
Thank you, Ramita. It's a great pleasure, and I should thank the organizer and Inari also for the opportunity to come and give this talk. Come and give this talk is a little misnomer because I've gone nowhere and still giving the talk. But I do miss standing in front of the crowd and students interacting, especially your department where I visited before, and it's always very lively. So let us sort of start what I want to share with you today a little bit about the journey of the last almost 10 years now, which is to understand cell membrane homeostasis using fluctuations. This is one part of the lab which works where we want to understand membrane homeostasis using fluctuations. The other in the other part we usually we focus on things like actin cytoskeletal remodeling in membranes and the cells homeostasis, which is, I mean, I'll focus on some parts of the work. So let us just get through the initial terminologies, which is first thing that comes to mind after hearing the title is what do you mean by membrane homeostasis? Uh, the very hypothesis which we are trying to test, and actually it has been tested by many labs by now, and a lot of the insights have come from the last let's say 10 or 15 years work, not 10 but let's say 15 or 20 years work, where uh, the 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 hypothesis is something that is not just about the overall state of the membrane, but the state of membranes mechanical mechanics, the membrane's mechanics. It is not something which is completely delinked from its biological state, and that is where the way homeostasis is looked at in this, like in the kind of work that I'm talking about, becomes relevant. So membrane homeostasis, when I say membrane, what I mean is membraning the state of tension of the membrane at a particular level. Why is this? How is it biologically connected at all? And a simple figure is given here for everyone who has done cell biology. You know that the membrane has a lot to do. It's the barrier between the inside and the outside of the cell, and therefore has to regulate what comes in and what goes out. And not only that, it takes part in cell shape changes, like certain ruffles are shown here, which is what is thrown out when cells move to feel the world. There are filopodia like structures, microvilli. These are uh, protrusions from the membrane, again needing a lot of membrane deformation. And of course, things like endocytosis, exocytosis, and processes which continuously keep changing what membrane is there on at the cell at any particular position. It is a continuous flow of membrane in going from the plasma membrane to the inside and then again coming back, which has to be regulated, keeping the cell surface area into account. The whole surface area of the cell is very slowly increasing as it is going through the cell cycle, the cell volume which is increasing. So membrane homeostasis is linked with tension by certain mechanisms. Now, which are the mechanisms which we started off, or which comes to your mind, or in the hypothesis of surface area regulation and membrane homeostasis, which is posed, are mainly things that eat away membrane from the plasma membrane, like removal of membrane, which happens by the process of endocytosis, or addition of membrane to the plasma membrane, which happens in recycling and exocytosis. These are two processes which are directly and most thought to be most relevant in membranes homeostasis or membrane tension regulation. So now, I did not introduce the term tension, and I will, but why is it again relevant? It is relevant because the state of tension regulates in itself the process of endocytosis and exocytosis. Just to give in some example, and not just these processes, but many other processes, like opening of ion channels. There are stretch responsive ion channels. Organizing the front of the cell when the cell has to move. So tension has its own role in for the cell to function properly, and tension needs to be regulated, because otherwise, uh, if there are inputs on the cell and the tension is changed and it is not regulated back, the functions of the cells will be compromised. So the idea here in this work, which I'm going to talk about, is to understand these mechanisms of membrane homeostasis beyond what is usually believed. Like, okay, this must be increasing tension. So for example, I've started here with some state of tension. The state of tension can be visualized, imagined in mind by just drawing a close microscopic view of the membrane and the membrane's undulations. So let's say this is the normal one. What do I mean when I say the tension increases? Here, what I mean is, when I say tension here, I'm really talking about surface tension. So I'm saying that the surface has gotten smoothened out, so that it is more difficult now. It is resisting any more change in tension, in change in surface area, and so the tension is actually increased here. While if I do something and it becomes more floppy and not as more taut, then the tension is actually decreasing. So what is there in the field is, for regulating tension, bringing back to the original state or maintaining homeostasis. There are mechanisms like endocytosis, which will take away membrane, and exocytosis or recycling will bring back membrane. And so we started trying to understand these things a little more deeper, and asking questions like, okay, so now for the main measurement of tension is measurement of force required to pull out nanotethers from cells. I've not put them in these slides, but you can, I have, in fact, a little later, you will see that, you can basically come with optically trapped bead, pull a nanotether out of the cell, and you measure a tension. However, in the lab, what we felt is that we should look a little more closer. As you can see here, this is sort of a measurement at this point, while the cell has very, very different kind of structures all over the cell. And but I'm measuring only at one point. So what we wanted to do is to first, even if we want to try to understand tension regulation, we wanted to come up with a way where we can map tension or look at or make multi-point tension measurement. For that, we went back to our layman understanding of tension, of looking at fluctuations or undulations. These are fluctuations, we call them fluctuations because they're height fluctuations. So if you think of the membrane and think of the glass cover slip on which it is sitting, then the distance of the membrane from the glass cover slip is changing as you go at different locations. So that is spatial fluctuations. Or if you had to just look in time, this membrane would be doing jiggling. Okay, it will be going up and down, up and down. And so these are fluctuations. So what we wanted to understand is that, this is a theoretical understanding or theoretical picture of the membrane. Is it something that can be measured in such a complicated structure? So people have measured using fluctuations in much more simpler systems like vesicles and even RBCs, which have more or less, although there is a layer of spectrin, you don't have very crazy kind of cytoskeleton, which is very heterogeneous. Crazy, what I mean here is heterogeneous, as in you see in other eukaryotics. So that those were the sort of basic first tool preparing time where we wanted to understand if fluctuations can be measured, if fluctuations are regulated, can we really derive tension maps from them, and then what I'm going to talk about today is about how it affects the cell in different ways. So the different arms of this work, what you see in the center, of course, is, I'll come to it, but this is sort of a color map of the cell's fluctuations. Ignore the black, this is something that we can't analyze, but otherwise you see there's a lot of heterogeneity. What we wanted to understand is, what is this heterogeneity? When I showed this cartoon of how it is usually measured, and the belief was always that, tension is something which is equilibrated really fast, so you shouldn't measure any different tension if you measure it again and again, or if you measure it in a different place. And we wanted to see a snapshot and understand the heterogeneity. We wanted to understand how a simple vesicle versus a cell, or even a dead cell versus a live cell with a lot of activity in it, with the underlying cytoskeleton, with pushes and pulls beyond what thermal background, bombardment by water does. How does that affect? Does it really change the characteristics of the fluctuations? Does it affect? What role does it have in regulating the fluctuations and in regulating tension? And as I said, that endocytosis is one of the most important mechanisms believed to regulate tension. We want to understand this. And then these three are about its functional roles in the cell's different processes. For example, if I have time, I'll go through cell-cell fusion, which is an important step in myogenesis, which is when muscle cells fuse together and form muscle fibers, in giving cells mechanoprotection and even cell migration. These are things that I will not touch upon at all, but I'll start with our understanding of activity, understanding heterogeneity, and we'll bring in endocytosis as and when required, and see if there is time I'll talk about cell fusion. So the system, just to introduce it, as just already introduced, is the cell, and I'm talking about tension. And I just sort of explained that it is. You can think of it as a tension. Of course, there are various ways of defining tension, and I'm not talking about the really intrinsic tension, which is due to which would increase if the lipid to lipid distance increased. I'm talking about the mechanical tension or the surface tension here, which is something you can imagine as if there is an imaginary frame around any patch of membrane, and the way the membrane is behaving as if this frame is at under attention, is giving attention in this direction. Hmm? So all its properties of fluctuations would be defined not just by this tension, of course, but also by its other properties like its bending rigidity, the viscosity of the media which surrounds it, the temperature. Okay, these are other, everything together will decide the fluctuations. And what we want to do is to use just this measurement of the fluctuations of the height. And what I mean by height, as I already told you, is a height from the cover slip from where it is. So this is a rigid, this is not changing its location, but this guy is changing its location. If you go in space or if you wait in time, this is going to fluctuate, and from that we want to understand the tension. Okay, there are various other parameters which are just sort of thrown here just to show you that it is not just tension which determines anything in the cell. There are all many other parameters or forces, kinds of forces which affect the functioning of the cell. Just to give you a sort of a disclaimer that while I will be talking about membrane tension, membrane surface tension, or fluctuation tension, bring it to the forefront to try to say that this is important or this does this or that in the cell, remember that all the other things are also there. Now, this one I've already talked about, where it shows different the heterogeneity in the structure of the cell and usual ways of measurement, where either you pull a tube and you find the tension out from the force, or you do interferometry at one single point. Or there are other measurements possible which are more invasive in nature. This is invasive, this is not invasive, but there are more invasive measurements possible, but they really work not on cells which are adhered like this, but cells which are floating and suspended, or best used of these techniques of like micropipette shown here are on vesicles. So we've gone from this to try to understand in cells, and as I said, we use the height fluctuation to come up with our understanding of membrane tension, and in the process have to do something with the fluctuations. So we do different kinds of analysis of the fluctuations. What I mean by fluctuations, so if you want to understand or measure the height, how do you do it? So we as was also talked about in the introduction, what we do is do interferometry or interference reflection microscopy, where the cell, now it's not like a bright field or fluorescent image of a cell which we're usually used to, but here we see black and white, the cell in black and white, and what we're really seeing is the lower basal membrane of the cell, where a very dark patch is a region which is very close to the cover slip, and something which is gray is slightly away from the cover slips. Okay, so the intensity of light tells you how far you are away from the cover slip. This is how you can sort of think about it. We don't have time to go into the details, but this is what, and this is calibrated, where, as I just said, I just talked about this arm, but it's not so simple. The intensity increases as you go away and away from the cover slip, but after a time it again comes down and again goes up and again, so this keeps on happening. Now we use beads which have exactly a circle shape underneath in the cross section. These are bought from NIST, which is the standard where you get all these standards, and this is a very, very well defined radius and curvature. You can trust them, and we use them and the cell together for our calibration, so that from the intensity we can get back the height. Now, as we get back the height in a relative fashion, so we don't know the exact, but the relative height, so that any change in height we can measure exactly. So whether it is the change in height as you go from one place to another place, or if you wait in time, you can measure exactly. Okay. So when we do that, either we get this kind of a map of the height in a small regions where we trust our measurements, or we just wait in one pixel and get the fluctuations. Using these, we derived various things like the standard deviation of these fluctuations as we go along in space, or if we move see in time, it's fluctuating, and get the distribution, get the standard deviation. Then we do correlations, and then we, as I already showed, we do the power spectra analysis, which is the final. If you want to get, these are all direct measurements, standard deviation of the amplitude of the fluctuations are direct measurements, but we do some indirect extraction of tension from the frequency distribution of the fluctuations, which is shown here. We fit it with the known model which I showed here. right? This is a model where this is the expectation of how the power spectra should look if you had a membrane of certain tension, of certain bending rigidity, with certain effective viscosity outside. Okay, once we do that, then what I've tried to show here is we measure these numbers for different little patches. Either we do that or we make maps of each of these things. And our initial work has shown that the numbers are more or less in the range of values reported. They come with great error bars because every system is different. Every system, when worked on again and again, there's slight difference. But what you have is is order of magnitude comparison done here, and this is sort of very well fits with the kind of numbers that are there in the literature. It just tells us that you can really actually measure surface tension in eukaryotic cells, which are otherwise extremely complicated and noisy, and people have tried to avoid them. Of course, there are limitations of this, and we see more and more the basically, and we are this is something I'll not talk about in this talk, but we're really trying to understand if the kind of model we apply to extract tension is a very non-active model while we have activity in the cell, and how do you really account for that? We have accounted for that in a way where we look at an active factor in the cell, which you can see here as this multiplying factor A. They almost assume that the whole cell is almost at a higher temperature. At all frequencies, the fluctuations are high. So I will first talk about this whole thing about activity. Does a live cell have special characteristics in its fluctuation? And what are the differences? Here, what I just talked about is also sort of shown here, where the power spectra of the live and the ATP depleted cells, cells from which ATP has been depleted to a great extent, if not completely, we see that the fluctuations actually reduce, and in the frequency range that we are seeing, it's more or less not dependent on frequency. It is frequency independent reduction of fluctuation. There is a slight frequency dependence, but it's not as bad as you would imagine if you had a much longer, if your whole measurement is dependent on a much longer or a much wider frequency distribution. Then you will really have to use a frequency dependent lowering of fluctuations and activity factor, which should be ideally frequency dependent. That is something that we are working on. But what are the take home messages here? The take home message is expected. It is intuitively clear that ATP depletion actually, as you can see, it is the fluctuations or the relative height here is coded in color. So these are controls typical for patches from the cell. Now, when you ATP deplete, all of them become more blue. Or staurosporin, which induces apoptosis, also becomes blue. Or fixed cells, also blue. All these little patches are also not there. You see, it's more uniform, more less fluctuating. So the fluctuations are dampened, but are there any particular signatures of it? Well, we see that one, just qualitatively, what we see is that there are certain fluctuations which build up and then go away. So you see the fluctuation is building up and going up. And these are sort of they're second time scale. So the imaging is done at 50 milliseconds and goes on for around 100 seconds or 80 seconds. But these things we see, the building up of fluctuations going away, are of the order of seconds. The going away is 0.5 second in its time scale, and going up is 3 seconds. When we do the autocorrelation, this is qualitative, but then we throw all the data and just close our eyes and just do the analysis in every region which matters, which is good, which is analyzable, and find the different time scales of the autocorrelation. Autocorrelation basically looks like this. You can fit and find time scale out of it, and when you plot distribution of these time scales, we see that there is a higher. So this red is the ATP depleted, and the black is the control or with ATP, and there is a more fluctuations in a particular time scale, which is seen in these live cells, and they're also not there. This is the fraction of curves. So this is about the time scale, but we also see these bumps, little bumps in the fluctuations, in the autocorrelation of the fluctuations, which are more also prominent in the live cells and not in the ATP depleted, and not in spheres of membrane which are extracted from the cell. So these are called plasma membrane spheres, and we don't see that in. So these look like that active fluctuations are not just perhaps an increase in temperature, but in certain specific regions you do see the effect of activity by by bursts of fluctuations and so on. But this is about the fluctuation. What does it do to the tension? On measurement of tension on those regions where there is nothing crazy going on, what we see is that overall there is an increase in tension on ATP depletion. This was a surprise because for long of the kind of measurement which I said, people have actually measured a reduction in tension. However, we see an increase in tension, and having done other studies which we're not going to right now, but any kind of perturbation to the membrane or to the cytoskeleton looks like increases the tension. Okay, it seems like the cell in its active and live and wild type and control condition is trying to maintain the tension in a lowered state. So job of activity and all the things that the cell is doing is actively trying to keep the membrane tension lowered. This is a little surprising, but this is what we find. So I'll take you okay, this is, I think, right now, since we don't have time, I don't want to go into this active thing. But this, while I just said that activity tries to reduce tension, this is context dependent. I'm only trying to talk about when I all the experiments that I talk about right now will be in the interphase cell cycle. Shown an example here where when sometimes the cell also wants to actively increase tension so that all the endocytotic and membrane activities are slowed down. And this is, for example, in the mitotic cell, where people have already published long back that mitotic cells have increased tension. When we look at with our way of measuring tension, we indeed see that mitotic cells have increased tension. And this thing is sort of seen in different kind of cell lines. Whatever we have tried to say, at least in 3 to 4 different cell lines, we see this to be true. So now I'm going to talk about how is activity related or comes in when now whatever I've done now so far, I've not let membrane homeostasis, I'm not following membrane homeostasis. I've just told you what is required to maintain homeostasis. But if I perturb homeostasis without perturbing the cell in a big way, like let's say actin depletion or whatever, let's do normal, do I see any okay, do I see the, can I follow homeostasis, and can I find out some rules of homeostasis? This is just to give you a break from the fluctuation. This is a deadhering cell in which the cytoskeleton has been marked, and you see that there's a initial, there is a crunching, there's a myosin based crunching of the cell membrane. The whole cell's spread area actually reduces once the deadhesion starts, and the actin is remodeled. Fine. And the cell actually goes to a smaller adhesion area and stabilizes there. In this background, what is happening to the membrane? So what I've done now, I've done something to take the cell away from its homeostatic state, right? And what did I do? I de-adhered the cell. And you can imagine in your mind that the membrane was flat, and then you're suddenly reducing the spread area, and so there should be more undulations. Can we pick up this? Do we see a reduction in tension? Yes, we see a reduction in tension, as you see here. Okay, so in two different conditions of trypsin EDTA added, there is a reduction of tension, and then we wanted to understand what happens next. I mean, there is a reduction, but is there like regulation? So what we realize is if I do a slow deadhesion, then with every time point in which deadhesion happens, actually the regulation is also happening. So even to catch the tension reduction is very difficult. So what we did, we played around and we found that if we quickly deadhere, so that all these cellular processes, they have their own time scale, they cannot catch up yet, so then we can differentiate and find out the different phases. So here we've differentiated and the slow and the faster deadhesion, and You can see the time scale of deadhesion for the slow and fast is different, really different. Fast one is much faster deadhesion. And anyway, deadhesion has, we had to understand the data, we had to put them in different places, phases, that there is a, before you add any de-adhering agent, it is in certain spread area, the area of the cell. Just before the deadhesion, catapults, sudden change in, there is actually slow change, and this is the phase one. Phase two, when you have sudden decrease, exponential decrease in the spread area, and then there is a saturation. Okay, so these are the CP1, 2, P3, whatever we get, we put them in the, whichever phases are accessible, we put them in the different baskets, and then we start analyzing our data. Here is a map of fluctuations, not tension yet, and you can see that the fluctuations build up and then finally reduce. So here you see more green here, and then there's the green here is actually less. So this is what, when you start quantifying, you start to ask questions, that is there a change in tension, is it ATP dependent, is it endocytosis dependent, which pathways it is dependent, what is it really, the whole endocytotic process is important, or for certain particular legs of endocytosis is important. This is what I'm going to talk about. First thing we understand is, so you see, this is the control. Okay, this is the ATP depleted cell. Oh God, what happened? Yeah, fine. We start with ATP. What happens when you do ATP depletion versus when you don't do ATP depletion and have the normal cell, versus when you inhibit all these endocytotic pathways? So when you do ATP, so I think I have sort of messed up my slide ordering a little bit. So as I said, we always will show you both direct measurements and indirect, because one can always ask if this is active, and you're looking at using an equilibrium model to extract tension, how good is this? So whatever we see in tension, we actually see its reflection in the fluctuations. For example, here you see that in the control cells and you deadhere, the fluctuations go up and then start coming down, right? So tension here goes down and then sort of saturates, tries to saturate. It's very difficult to catch it in the control, but in the ATP depleted case, and this I'm not even putting in phases, I'm actually following it in time. In the ATP depletion, you see that nice fall and then coming back, and it was a surprise. We thought that since endocytosis process is one of the most important in tension regulation, perhaps ATP depletion is going to kill all the different processes and it shouldn't increase. Maybe it can decrease because unpinning can change tension. Unpinning means the membrane is attached to the substrate, so that will also create an enhanced tension. And when it deadhere, perhaps that's why it should decrease. But what we see is that ATP depletion, you can see here I already told you fluctuations are already low in ATP depletion, but when you deadhere, fluctuations still from wherever they are, they increase and then they decrease. So there is a regulation that's still happening in the ATP depleted cell when compared to a control cell. This is the surprise number one. Then this is the next thing which we did, which was look at both. Okay, this is a mistake here. This is actually not really click-geek inhibited cell. This is a Dynamine inhibited cell. As you see here, Dynamine is inhibited by a drug called Dinosaur. So we are looking at clathrin-mediated endocytosis. What if we stop clathrin-mediated endocytosis? And then we'll look at click-geek mediated, which is a click-geek pathway, which is really implicated in tension regulation. So here what we see is that, again, well, the drop I expect, because it is like a trigger. It is the input. Input is still there. Deadhesion is happening, so it'll reduce. But it is still coming back, and it is not just coming back, it is going more than the initial tension. So look at the control drops, but by the time that we're looking at, it's really not going back higher than this tension. It does, and I'm not included this in this presentation in view of the time we have. But if you look a little longer, like if you wait for 1 hour, actually the tension doesn't go back to the original state, it goes higher than the original state, which we believe is because the new tension should be dependent on the state of the cytoskeleton. The cytoskeleton is weaker, so more load comes on the membrane has to take it, and it has an increased tension. But those are things that will not get into now. But in the same time scale that we're looking at, where by this time the tension has not gone up, is that the right way to talk about it? We'll discuss it soon. But when you block endocytosis, and blocking endocytosis in which step? When you're blocking Dynamine dependent endocytosis, not just the clathrin. So in this slide, which is wrongly labeled, you're putting dinosaur, which blocks all Dynamine dependent endocytosis. So Dynamine is important in that place of endocytosis when the pit has formed, but the pit cannot be now pinched off and taken in. Remember that, keep it in your mind, and we'll come back to it. So there is a decrease and there is an increase, and not just increase, is more than the initial increase. Same about click pathway. When I inhibit click pathway with an inhibitor which also inhibits the pathway by stopping the pinching off, the actin remodeling based pinching off of the endocytotic tubules that are formed. The click pathway is known for forming various kind of things, including tubules, in the initiation of the endocytosis. We see that when you block that there is an increase, but there is no regulation. I mean, it's not even like it. I mean, with respect to the wild type or the control where nothing has been changed, it is much higher. Okay, so it's even higher than the initial tension. Now, this led us to, so what did we see? ATP depletion doesn't stop the pulling back. Endocytosis blocking doesn't stop, of two different endocytosis doesn't stop the pulling back. So we just stepped back and checked again our old results with what happens when you put Shiga toxin on cells. This is a toxin which is taken in again by the, of course, by FME pathway, which is cholesterol dependent and has tubules formed which are endocytosed off. And here what we had seen is that when you have Shiga toxin, after incubating it for Shiga toxin, the membrane actually increases its tension. Very slight increase, but there is a this is reproducible and robust and statistically significant increase of tension in cells. When tested on ATP depletion, tension increase was still there, or not just still there, it is actually in more tension increase. Now, the beauty of this experiment is we have fluorescently labeled Shiga toxin, so we can actually visualize it, and people have already published this that even in ATP depleted cells, the toxin makes the tubules. It is the pinching part which is defective when you ATP deplete cells, cannot go on because it is again Dynamine dependent, and Dynamine is active. It depends on the activity of the cell, the metabolic state of the cell, otherwise it can't work if it is ATP depleted. So you do see tubulated structures in ATP depleted cells, which means there are tubules formed, and in that state it is getting stuck. It is not going through the whole process of endocytosis. So, and Shiga toxin was increasing tension. The two different triggers I'm here discussing. One, where the cell has to regulate back its tension, that is why it wants to increase the tension. And here you have been treated with Shiga toxin, and the toxin is taken in. It is initiating some processes which are eating away the membrane, so the tension has to increase. So what we realize from this experiment, where again I'm talking about the toxin experiment, is possibly that it is not the whole process of endocytosis which is essential for the increase in tension. Even at the very beginning, for the toxins, we have, for example, looked at the enrichment of the toxin and the standard deviation in time, which is the temporal fluctuations denoted by the pink color going up and down, and the spatial, which is we are trying to draw by just the lines. You see the spatial is this much. When you have toxins and they form clusters, this is a speculation, so therefore all probable here. What is plotted here is representative of a few cells, but what we believe is that here the tension standard deviation decreases where the clusters are really stopping the fluctuations from happening. Oh god. And the spatial is also affected because now you will have rigid things which now can't really bend in this local length scale, and therefore both the standard deviation and space increases. What you see is by this time tension also has increased. You see there is an increase in the tension by this time. There is, of course, this takes time to get statistically significant, but it has started increasing. Definitely with this time where the tubules have formed, the tension. So if we are not very sure if clustering can increase tension, but it looks like tubulation itself and not the process of uh, pinching off. Tubulation is enough for tension increase, and this is more clear because we have already shown that in ATP depleted cells the tension increases. So looking from this, what it looks like is that there is a passive way of tension pulling back, right? Because even without ATP, I got the tension pulling back. Pulling back meaning there is a dip, and this going back up is passive, is not ATP dependent, does not need ATP. Does it need anything at all? And we show that this is definitely cholesterol dependent. Same experiment, ATP depletion, but cholesterol also depleted. You see that it does come down. As I said, dehydration is a trigger, whatever you are, whoever you are, ATP depleted or not, cholesterol depleted or not, it doesn't come back. It cannot, the passive regulation of tension is not happening. Now, a lot of work has already been done in the field where these tubules formation are understood to be cholesterol dependent, and there are domains like raft like domains which can at a lower tension form tubules. Looks like this is something that is happening in a very general way in um, and essential for the first step in tension regulation by endocytosis. So of course these platforms are there, their main role is endocytosis. Even if you stop the process of endocytosis, block the process of endocytosis, that they are present. And it looks like cholesterol is very important for the passive pulling back of tension. When you have of course when you have clathrin present and clathrin coated pits can form, that is also increasing the tension. But here, that cannot really happen in the in an ATP independent fashion. So when in the ATP independent part of the tension regulation is cholesterol dependent, is what our understanding from this part is. We actually also try to look at it, by really looking at the endosomes, because if we are saying endocytosis is increasing, decreasing, what we are really trying to say here is at one part, this part, there is a decrease, and because of the decrease, endocytosis is definitely increasing, or the pits are forming. So in the live cell, then what is happening is more pits are forming. That is our speculation. More pits are forming, and why is it not dipping so much? Because hand in hand, it's also recycled back, right? So what do you expect is that you expect, if really this was to be true, you expect an increase in the fraction of endosomes which are early endosomes, maybe also the recycling endosomes. Okay, so what we see here is, in fact, your this is the so the RAP4 marks the early endosome as well as recycling endosome. RAP5 marks the early endosome. So RAP5 early endosomes actually increases more in the first step, less in the second step. Expected, because by the time the second step, which is in for them the 10 minutes, tension has already started increasing. This tension is no more so low, so tension increase is not so important. So the rate of tension increase is less here, and so your endocytotic number of endosomes is also increase of number of endosomes is less. Sometimes in this case it is NS. What happens to RAP4? RAP4 here, you see that RAP4 is also increasing, but this is increasing transient, after which it starts decreasing. So which means what? RAP4 is the label of the early endosome, recycling endosome. So once it's recycled, there is no more RAP4. So you have to count both things, that there are more RAP4 coming in, and there is a recycling of the RAP4. So there is, which reduces the amount of RAP4. So RAP4 goes through increase and a decrease. Sometimes you don't see a decrease. That is our speculation. These things still need to be, really pinned down. And we see that in the same cells, when we measure RAP5 and RAP4, Here you see, for example, this is a slow thing where you see RAP5 increasing, and I can't yet see the saturation or decrease. By this, in this experiment, you see an increase and a decrease, and RAP4, perhaps you miss the increase, you start seeing the decrease. So both these experiments tell us that the hypothesis that endocytosis, the alluring of tension, induces endocytosis, and this is not surprising. This is believed in the field. We just wanted to see in our own hands, in our own time scales, because there are no studies which follow it in time and show how, in what steps is tension regulated. Okay, so what we started off with, we said that, in endocytosis is important for bringing the tension higher up when there is a tension reduction, and recycling is important when this. What we're trying to say is that it's not so simple. If you reduce tension, then there is a passive formation of these pits and tubes, hmm, depending on if you are ATP depleted or not. If you are ATP depleted, only the tubes form, otherwise your endocytotic pits also start forming, but the formation already increases the tension. It is the recycling which is important for, and recycling is a part of the whole process of endocytosis, which is important for regulating the tension back. Otherwise, it is a lot of uh, un, um unhindered increase of tension. This is what you see when you block endocytosis in some of the pathway. So there's so many of these are formed. There is no regulation of how many you are forming, and it keeps on forming, and you go higher than this tension. Only if you have the whole of endocytosis there, that you regulate it back to some level of tension. So it looks like it's not so simple that endocytosis helps in increasing tension. Endocytosis, it looks like, helps in regulating tension back. And this, so it has both these arms. So recycling and pinching off both are important, and not just pinching off. Formation of the new thing here is that formation of these structures, early endocytotic pits, are important for the tensions being pulled back when there is an increase in tension. Now, something very basic, and I said initially that, you do 1 point measurement, and then what is the use of multi-point measurement? Did it tell me anything? What I plotted here was all, numbers. I did not show you maps of tension. We have maps of tension, but it's not shown them yet. I'm going back a little bit in time in the way the work has progressed, and what I wanted to show you, and you saw that cholesterol was important in the passive increase of tension. Here, what I show is what happens when you deplete cholesterol. Acute cholesterol depletion from the membrane using an agent called Bidisha, which comes and sits on the membrane and extracts cholesterol. It's not affecting internal biogenesis of cholesterol. So not long term. It's a 1 hour application. So here using Philippine, we have labeled the cholesterol, and indeed it's going down. And you see the IRM, the interference reflection microscopy, shows a very different. You can see, anybody can see the difference. There's qualitatively different membrane undulations. Looks like they're flatter in certain places, and then they come down and they remain down for longer places. We also know, we cannot disregard this fact, we also know that the membrane actin attachment is also changed. However, we did not see any such huge patch formation of, so we know that this is not due to, although the membrane actin, here we have used turf imaging of a protein which binds the actin to the membrane. We don't see such huge patch formation as you see here in the membrane undulations. The correlative experiments have also been done, and they don't show they don't make ezrin or the membrane actin attachment to be important for the such geometry. It's more the, I think, the way the membrane composition is changed. What we show here is now a tension map. So now we are coming to the usefulness of making multi-point measurements. I'll not take much time, maybe 10 more, 6 more minutes. What we see is that yes, tension, there is a non-uniformity of tension, but there are more pockets formed, if you can see the pockets here, when there is cholesterol depletion. And not just this heterogeneity is not just true in tension, but we've shown that it is also there in the standard deviation of or the heterogeneity, or the intracellular heterogeneity of the spatial fluctuations amplitude. The amplitude of spatial fluctuation, if you measure in different places, and then you see intracellular heterogeneity, and you take this in many cells, you see that cholesterol depletion is actually increasing the intracellular heterogeneity. So the membrane is now becoming more patchy in in terms of tension. And maybe that is why when your cholesterol deplete and then you de-adhere, it cannot form these tubes because somewhere there is high tension, somewhere there is low tension. Only when you have a mixture, maybe those platforms are all gone, and even the tension is not in a place where it can quickly form these tubes. What is interesting is that patches of tension, so we are therefore presenting evidence of heterogeneous tension. There are measurements of optical trap based tube pulling from 2 points, and there's a 2018 Cell paper we published in 2019 where people have shown, the group has shown that, or the data implies that tension is not flowing quickly. So if tension cannot flow quickly, tension cannot equilibrate quickly, you do have a chance of heterogeneities propping up because cell is doing different things in different places. So here you can see an heterogeneous pocket which is gone from you. We thought, and look at the tension increases, you remove Absorption, tension again starts going down. But what we see here is the recovery. So again, we're looking at homeostasis here. The recovery is a slow, and the paper I just talked about gives you, if I these are very rough estimates, we have not yet communicated this, so these are sort of still new, where the if you can think of a diffusion constant of the tensions equilibration, what is proposed in the literature saying that it is really really slow is 0.04. We measure much less than that because when we follow this patch on the same cell as ambidacity is taken out so that it slowly recovers, it's not just, so if it was to flow fast, even before recovery it would flow, but it looks like it doesn't flow so fast. Not just flows so fast, it's perhaps it recovers not by flowing, although this is the effective d put in here, it recovers by remodeling of the membrane. And this you can see is that what has been shown here is that it's the number of patches actually increases, and it's counterintuitive also. It's ns, you can, it's the trend is there, it's increasing, but it is, when you think about it, what is happening is from bigger patches you're making smaller patches which are finally going. Okay. And does it have any, like, you are just, we are just digging deep and giving you some information, does it really have any implication in the cell's health? And it does, where what you see is that here cells going through a little hyposhock, you give a little mechanical stress on the cell by giving a hypoosmotic shock. Cells which are normal, they are retaining their internal fluorescence, which is some dye which is put in, and they're not breaking. So very few of them actually burst. But the cells which are treated with or cholesterol depleted, you can, when the movie runs again, you will see that are just plopping, pop, 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 gone. They're really defective in their membrane. So heterogeneous membrane tension is possible. It makes the cell really fragile. So coming to this in the next 2 or 3 minutes, I just want to show you that we also have hints of tension being important in cell-cell fusion. Okay, so people who are interested in the lab, if I'm hopes there are some master students who are looking at it, you know, it's a very biophysics kind of lab, and if, but we are trying to do more, you know, things which are processes which are relevant and connected to sort of processes like differentiation. Here we see that so again we try to see this from the angle of spatial heterogeneity. We see that in fact the spatial heterogeneity in myotubes, so these are now myotubes and not just myoblasts which, you know, fuse and form myotubes, these are myotubes, have higher tension and higher heterogeneity of tension. Okay, this was like an effect of differentiation on tension. We also asked we also try to understand the question: can we try to see if tension has any role in defining the fate of the cells? Because we know that when we put in differentiation mediums and see that some cells differentiate and many don't differentiate, they stay in the undifferentiated form. So is it to do with the initial tension they had? And this is where we started following cells. Following cells, when we follow cells through differentiation, we see that some cells differentiate. And the end of 5 days, 4 days, we fix the cell. This is still here, it is live, and then we fix the cell and do immunofluorescence, and you see the MF20 is really high than all the surrounding cells, which means the cell is differentiated. While another cell which was flown taken through doesn't have them, so it does not differentiate. So now since I've done live imaging and I went to the same cell and image, not going to the details of how we do that, we can find out what happens to the tension. Typical for a particular cell, you see, shows a drop in tension before it rises again, and after differentiation, of course, it is increasing. While the cell which is remains undifferentiated doesn't necessarily do this. This cell cannot really reduce its tension. Now we look at this. Now we go back in time. We know the final fate here. We go back in time, and when we did this experiment, we didn't know what will happen. But when we have done it, we know we can go back in time and say that, acha, this cell was different. This cell is a cell which will finally differentiate. This cell is a cell which will not finally remains undifferentiated. Then we pool them and then we measure their tension and see what is the difference. Is there a difference between these cells in this time point? And looks like there is. There is. And this is happening right 2 hours after putting in the differentiation medium. This makes us feel that either tension is somehow important in defining the fate of the differentiation. However, what I've not put in here, this is something a work we are also doing with Rumi Day. She's a professor in the physics department. It may not be so simple that a particular cell's tension is important. We believe that it is the interacting cell tension which is dependent, which defines the differentiation. When there are two cells, one with a lower and a higher tension, then they fruitfully differentiate and give rise to a differentiated cell which has a higher tension. If you just put this model there, we feel that we can explain this pooling together of cells which finally differentiate and finally undifferentiate, showing you the difference in tension. Okay, beyond this, we also try to understand the molecular players involved. And myomerger is a molecule or a protein which is a small ectopic domain and is on the membrane, on the plasma membrane. When it localizes on the plasma membrane, it has a small ectopic domain and it's known to stress the membrane wherever they are, wherever it is. If we've and it is shown in like 2017, it has come out to be a very important molecule, and people have shown that cells, even like cells like CHO, which don't usually have myomerger, if you put in myomerger, these cells will start fusing. Now there is of course also a recent paper which show that myomerger is not necessarily always, I mean, increasing concentration will not increase fusion of cells. There is a band of concentration in which it is important. Okay, above that it will not help fusion. Below that it will not help fusion. We try to understand. We're trying to understand the effect of myomerger on tension, because we see that tension has an effect in the process of differentiation, not just as an effect of differentiation, but also may be important in deciding the fate of a cell, or deciding whether a cell will differentiate or not. So we looked at myomerger, and interestingly, we see that myomerger is first going down before it goes up. Okay, this is, I think, to my knowledge, this is one of the first careful work of measuring myomerger at the surface. We're not looking at whole cell myomerger. We're looking at the myomerger. This is done with total internal reflection microscopy and immunofluorescence, measuring myomerger at the membrane, very close to the membrane. We see that there is actually a drop, and then there is an increase in tension, an increase in the myomerger, and this is very close to what you see for cells which undergo differentiation. This pattern is same for the tension in cells which go through differentiation. This brings us to the sort of the end of the story here. I'm not talking about we are doing experiments which are correlative microscopy, where we can measure the tension map of a particular cell and put myomerger map on top of it and then see the correlation. This is a correlation of the tension and the myomerger in time across a whole population of cells. Roughly how they go. Looks like they go hand in hand. But when we look at in the cell and try to see spatial correlation, like in this space, if there is more myomerger, what is the tension? Is it always higher? We see it is not true. In fact, it is again something like that has multiple arms. So if very low is high tension, then again as you start increasing myomerger, there's a decrease in tension. You start further increasing myomerger, there's an increase in tension. Okay, so myomerger can take you through different phases, which we are trying to pin down right now, but we are this still evades us as to how myomerger can affect differentiation, if myomerger can affect differentiation through membrane tension. The work that is this one work which proposes a membrane curvature being changed by myomerger, which affects tension and which affects fusion, and we are trying to look at it from the perspective of tension. So let me conclude here before going and not go into other things. What we just said and the take-home message is that active fluctuations can be measured at the plasma membrane. Metabolically active cells are trying to keep the tension lowered. And this is not just seen in a normal cell and doing huge perturbations, but also seen in cells which are going through the process of de-adhesion. And here you see that if there is a live cell, then it goes tension is decreased, and then when it increases the tension, it does not increase tension unhindered. It tries to bring back tension. So it tries to keep the tension lowered at its set point. However, when you do the same thing with the ATP depleted condition, so ATP is not the tension, but you have still jabardasti lowered the tension by de-adhesion. Now it just goes up and it cannot be brought down. So it looks again and again that the main role of active cells is to keep the tension lowered, and even endocytosis, and that is the surprise. The sort of the surprise or a new thing here, because endocytosis is usually implicated in bringing up tension. Yes, it does bring up tension, but the whole process of endocytosis includes its recycling, and recycling is important for regulating tension at a particular level. And so therefore endocytotic blockers don't stop the increase but stop the regulation or homeostasis, or stop the proper membrane homeostasis. I've also shown you very quickly though how tension not only is affected by cell fusion but may also regulate cell fusion or decide cell fusion. So I would like to stop here, and 1 minute after 8, it's a bit long, I'm sorry. I have to acknowledge the work of the whole lab. These are people who really drive. There are many new people in the lab now. The work I mainly talked about was started by Aurikta's work and carried on by Tithi. Okay, I've missed Tanmoy's name here. I've shown you Aurikta, Tithi, and Tanmoy's work, and Madhura's work today, whose work was on myoblast fusion. And Alekhya, Upasana, Jivitesh are new in the lab, and I have many old students in the lab who are listed here. Rinku's work, both Aurikta and Rinku are now postdocs in different parts of the world. She's in Singapore, he's in Germany, and without them the lab wouldn't have started and carried on. Then I should acknowledge the funding from India Alliance TBT Welcome, and I've again forgotten one logo of Cefipra here. DST. These are funding agencies who have funded us, and collaborators Rajesh and other labs of DBS, Biocluster, CCAM for STED imaging, it's not included here, cells and reagents from Jayashri, Deepak and Ludger's lab, and various ongoing collaborations which keep us, motivated and going on despite what the COVID has really made us face. So, yeah, I think I'm done, and thank you very much for your attention and being around. Yeah.
Thank you, ma'am, for your wonderful and enriching lecture. So, we have few questions from
Okay.
our audience.
Yeah.
So, with your permission, can we go through them?
Please do.
Okay. Our first question from Akashlina Marik. Can
Hmm.
nutrients and other factors play a part in tension of gut cells?
Very interesting question. What we see is that nutrients, of course, play a part. For example, I've not shown you here, but say when we serum deplete cells, then the tension increases. But we are showing you in HeLa cells or CHO cells, the cell lines. Recently, we have started a collaborative work with Malancho's lab, Malancho Taz lab, who they work on stem cells, more primary cells, right? So we do see that tension again can increase on these stresses. And I don't want to really, we cannot yet talk about the results yet, but there is a high possibility. What happens in the gut when there are other cells around? How would nutrient really alter the tension if there are other factors which keep the tension still at a lower level? I cannot say that. So that is something that can be addressed, that if a cell in its isolation is affected by reduction of nutrients. But in the presence of others, are there other regulating factors from other cells in terms of secretions or whatever, which would try to keep it back? I don't know. Yeah, I think
Okay, ma'am.
that is one.
Okay. Our next question is from Shomojit Bishas. Can we
Hmm.
measure plasma membrane fluctuations in cancer
Hmm.
cells?
Yes. So basically, the HeLa cells that I showed are cell lines, cancerous cell lines. But any cell, and I mean, I didn't get into other things that we are trying with other people. Like we were looking at primary cells like PDCs and also immune cells like Jockat cells. Any cell which has a little bit of adhesion. So here you see the cell has very poor adhesion, right? But that is enough to measure tension. You need some bit of adhesion. So here, at this portion, the adhesion is very weak, and these are cell lines which are cancer. So you can measure in cancer.
Okay, ma'am.
Hmm.
Thank you. Our next question is from again Akashlina Marik.
Hmm.
She says, you mentioned cholesterol plays a role in tension.
Hmm.
Can dietary cholesterol, that is high fat diet, play a role in tension of gut cells and affect endocytosis or absorption?
Yes, it is possible. So we are. I mean, again, I have not worked on gut cells, but as I told you, the way we are cholesterol depleting is a very different, I mean, is through imbitacity. It's a molecule which takes away cholesterol. But what we are also doing in the lab with Ananya, whose name I also missed here, is using statins. Statins are drugs which usually people take which who have hypertension problems, and we've tried to look at how that and we see that different statins have different effects. Some statins don't have any effect, other have marginal effect in trying in this whole thing that I just showed here of this rupturing. Not as bad as ambidacity, so you have to understand this is really, really exaggerated so that you can really measure these effects. But there are effects. I'm sure it would be effective. I'm quite sure that the way we are seeing cholesterol's role in tension regulation, homeostasis of membrane homeostasis in different ways, not just in membrane rupture, but also this initial tension regulation, is extremely cholesterol dependent. So if one looks carefully, I'm sure one will find an effect.
Okay, ma'am. Thank you. Our next and probably the last question is from Bijoy Vishas.
Hmm.
He asked how membrane tension can affect a cell fate. And
Hmm.
his next question is there any role of membrane tension in maintenance of stem cell niche?
Hmm. We don't know stem cell niche, but I can tell you that. Let me take the first question first. What was the first question again? Cell fate,
How
right?
membrane tension
Huh,
has self
huh, huh.
cell affect cell
So,
fate?
yeah. So the fate. What I'm talking about fate here is that they both of them started at the same initial condition. They're the same plate. Actually no, different representations from different plate, but in the same plate we have some cells which are finally differentiating. For example, here this cell has differentiated, some single cell here has not differentiated. Hmm? So you see that this is what I mean by cell fate here. Fate mane, I have been given differentiation medium. Will I differentiate or not is my fate. Of course, it's really not. I mean, the fate is a very like what we are putting on it, it depends on your gene expression profile, perhaps, right? But there are certain stochastic players also. Like given input, the input here is the differentiation media. Maybe certain gene expression patterns are stochastic. You cannot really predict that which way will it go. Hmm? What we are trying to understand is that is tension playing a role in deciding, because there is a good amount of stochasticity or cell to cell difference in tension in a population. Hmm? And that is not only to do with its biochemical state, but also to do with things like how it is adhered, who it is adhered with, and so on and so forth. There's some bit of difference already. Can that really decide the fate? Now, it is possible. We still don't know the answer. We know that it does. It It is affecting. Why? Because if I pull all the cells which are finally differentiated versus all the cells which are finally undifferentiated, there is a statistically significant difference in tension between them, even at 2 hours. So they have a different tension trajectory well before differentiation. So differentiation is happening at 48 or 72 hours after addition. But even at 2 hours, there is a clear change, difference in the tension trajectory. How tension of that particular cell is changing in time is different already before differentiation. So which is why we believe is that tension has a role to play in deciding if the cell will differentiate or not. Maybe not alone, maybe with other factors, but it has a role. Second question was what? Just remind me. Hello?
Yes, ma'am.
What was the second
His
question?
next question was: is there any role of membrane tension in maintenance of substantialness?
Yeah, so see, I mean, our little bit of work together with Malancho and her student Ankita and Madhura here, we have we're really not looking at central niche, meaning we're not looking at the stemness, let's say, coupling it with gene expression patterns of those genes which tell you that if it's a stem cell or not. We're actually only taking stem cells and giving them different conditions because this is related to what happens if you inject stem cells at a place where you want it to play a therapeutic role. A change in temperature or stress. How does it affect? But there are other people who have worked on stemness and at least things like how nuclear shape is fluctuating. It is seen that stem cells have a more supple, more fluctuating, more dynamic state than once it is differentiated. It becomes more stiffer than once it's okay. But
Okay.
exactly membrane tension we've not looked. Okay,
Okay.
I think they were nice, interesting questions, and I should thank all of you for.
Uh, yes, ma'am. Thank you, ma'am. Our next and probably the last question is from
Okay, another.
Sulanjana Haldar.
Okay,
Uh, she asked, cholesterol depression increases membrane tension.
right.
Can patients of CVD could get heterogeneous advantage in that respect?
What do you mean by CBT? Hello,
Yes, ma'am. She asked for CVD.
CVT. What is
CVD.
CVT? CVD?
Yes,
Cardiovascular dilation. Is that what she means?
maybe she means that.
Hmm. No, I mean the question is very. See, we are trying to understand this from the aspect of we know that the cholesterol levels need to be proper at the membrane for the membrane's mechanical strength. But our cells, our heart doesn't go through hypoosmotic shock, but it does go through stretching and relaxing, stretching and relaxing, where it pumps. So there now, if you have a difference in cholesterol levels, either due to the disease or due to the drug that you're taking, how it affects the mechanics or the integrity of the membrane of these cells is something that would definitely have an effect.
Okay. Thank you, ma'am.
Hmm.
Uh, that is all the question we have. Thank you,
Hmm.
madam, for answering all the questions. Now,
Hmm.
on behalf of our convener, Professor Dr. N.R.A. Banerjee, and from
Hmm.
all members of the Ant Biology organizing team, I would like to thank our respected speaker, Dr. Bidisha Sinha, for she has joined us with her wonderful and enriching lecture, about which has enriched us a lot and to all our audiences. For
Thank
our
you.
viewers, thank you for joining us today. Joining us again on 20th November 2021 for the special lecture by Professor Nandan Shastri. Please subscribe our YouTube channel and check out our website www.zoology zoologyhub.org. Our talks will be archived in the YouTube channel so that you can check out them later. And thank you again for joining us. Take care, stay safe, and see you again. Thank you.
Thank you. Bye.